So the first talk is going to be a virtual talk on LeGenD or LeGenD. So it's like LeGenD. So it's LeGenD, determining glycoprofiles using an explainable AI leveraged model with lectin profiling. And this is by Haining Li. So Haining Li, who is at UCSD. It's actually going to. And so, and it's on Zoom, so I have to.
Hello, everyone. So can you guys hear me? Hello?
Okay, here we go. Haining, please take it away.
Okay. Thank you, Professor Laura Mahal, for introducing me. So can everyone hear me okay, right? Okay. So I'm grateful for having this opportunity to present my work at the GIA today. And I'm gonna present LeGenD, an explainable AI model that determines glycoprofiles with lectin profiling. So to start with, I hope we all know that glycosylation assembles glycan onto glycoproteins, and where glycans are fundamental biological polymers with diverse range and functions. So for our lab, glycosylation is interesting because most biologics or biosimilars are composed of glycoproteins. As such, their glycan composition and structures must be carefully characterized and controlled, since they can greatly impact the drug safety and efficiency. Therefore, glycosylation profiling is critical to biomedical research. So nowadays, there are already some analytical tools to measure glycans, such as high-performance liquid chromatography, mass spectrometry, lectin arrays, and etc. So these techniques have advanced to provide high-quality profiling, but still, they have some limitations. So usually, they are time-consuming, costly, and require specialized training to interpret the result. So considering the growing awareness of the importance of glycobiology research, there is a need for new techniques to quantify the glycan compositions. So while using the MS is highly suited for glycan analysis, we are also interested in harnessing other technologies that can easily provide information on glycan epitopes that are compatible with downstream technologies. So therefore, we aim to develop a computational tool to resolve glycan structures solely from the lectin binding data. So here, let me introduce lectins a bit. So lectins are carbohydrate binding proteins that recognize mono and oligosaccharide structures to measure glycan features. However, it is important to note that the lectin binding patterns cannot be connected to a single glycoprofile. For example, our MS glycoprofile of glycoengineered CHO-derived EPO showed that its simulated lectin profile can map back to many strikingly different MS glycoprofiles. Well, but the good news is that each cell constrains the glycoprofiles they produce based on the species, enzymes, monosaccharide availabilities, and etc. So with this information, the range of the possible glycoprofiles would be greatly reduced. So here we have hypothesized that using AI model could address this under-constraint challenge by learning the inherent constraints on the glycan patterns imposed by the cells and proteins. So we present our work called LeGenD glycoprofiling, which uses data-driven model to predict glycan structures using the lectin profiles. The workflow shown here can be summarized as: first, our glycoprotein of interest are profiled using assays with lectins that recognize the features of common glycan structures. And then second, the lectin binding patterns are then used to determine the complete glycan structures using artificial neural networks trained on the lectin data with known glycoprofiles. And lastly, we use the SHAP with to determine the important lectins. So taken together, LeGenD offers an alternative platform to easily decipher the comprehensive glycan structures with high throughput and cost-efficient lectin profiles. So while developing our tool, the first challenge we met is the lack of data. So our collaborator, Wahlberg Lab, generated 309 glycoproteins. However, the lectin binding patterns did not come along with this dataset. So we had to simulate their lectin profiles by multiplying a glycan feature matrix by a binding rule matrix, which was adapted from published lectin affinity factors. But still, it is imperative to test if the values are consistent with measured lectin data. So we used human IgG and vacuum B samples to validate this. So where we simulated their lectin profiles and compared the simulated values to their experimentally measured lectin bindings. And we observed a coherence between the data for each lectin, which gave us confidence in using these simulated lectin profiles to make up our training dataset. Next, we built a fully connected neural network model connecting the 8 lectin measurements with the 68 N-glycan structures from the 309 glycoprofiles. So the model was evaluated for its ability to predict the N-glycans from the enzyme-linked lectin acids-derived lectin profiles. So after the model is trained, we tested its performance with the IgG lectin profiles. As shown here in this bar plot, we have the red bars as the experimentally measured glycoprofiles, and the blue and orange bars are the glycoprofiles predicted by the model. So using our model, we could predict the identity and abundances of the IgG glycans. From this, we have achieved high accuracy in capturing the expressed glycans, especially these with a relative abundancy above 0.1. This is impressive given the large number of glycans that could be predicted and near infinite combination of glycan patterns that could be determined from a single lectin profile. And then lastly, we use the SHAP to interpret our model. So a brief introduction about SHAP here is that SHAP computes Shapley values to determine the most important features and their influence on the model prediction. As shown in this two-way model, we can see that the feature age had the most impact on the prediction since it got the largest SHAP value. So here, we performed the SHAP to interpret how our model made its prediction for an IgG sample. By summing up the SHAP values towards the observed IgG glycans, we found that SHAP revealed that LCA and MAL1 were the most important lectins, with a total SHAP value of 0.4 and 0.35 above the average prediction. And in contrast, RCA, SNA, and PHAE, while still important, contribute less than 0.2. And this analysis also underscored the importance of DSL and PHAL with the lowest SHAP value. In conclusion, so in this study, we have presented LeGenD, a method to map lectin binding profiles to predict the identities and quantities of each glycans in a sample. So we demonstrated that this approach is made feasible after a model is trained with diverse glycoproteins, suggesting that the AI model captures the inherent relationships between the glycans determined by the cells. And then lectin also presents as an alternative approach for glycoprofiling to complement existing methods, that we use the AI with a large Glycomics training dataset and successfully compute a glycan profile based on lectin binding profile. And lastly, SHAP have provided further insights into the lectin importances, which could help us to shape the lectin panels for future work. So in summary, LeGenD has provided a complementary concept that with further development could aid in the proliferation of glycoanalytics in the routine biological research. And here I want to thank people from our lab for their help along with my work. So special thanks to my professor, Nathan Lewis, and our scientist, Austin Chen. This work could not be done without their guidance. And also special thanks to Angelo, the postdoc in our lab, who was mainly in charge of the lectin assays experiments. And thanks for Liu and Albert for assisting me doing this research. And also thanks for the people who did the glycoprofiling for us. And also thank you all for listening. This is my work. So I'm open to take any questions.
Great. Thank you so much, Haining. That's really interesting work. Are there questions? We have a few minutes. So I have a question. So in this, so thinking about lectin profiles, so I've got a couple of questions. One, is this mainly based on ELA data with lectin assays versus sort of, I'm just thinking about the different methods that one uses lectins in. Like, is this program sort of really intended for people using ELISA-like, ELA assays?
Thank you. That's a great question. Yes, so for now, the current stage, we are just mainly taking in the ELA-based lectin assays as our input.
And next, yeah, so I was going to ask one more question briefly, which is, so do you think, so this is interesting for single glycoproteins, do you think it will be applicable to more complex samples?
What do you mean by more complex sample?
Samples, for example, a cell surface, so the membrane proteins of a cell surface, or the lysates of a cancer cell, rather than a single protein. I mean, in the case of IgG, there's a very limited glycome, right? So the correlate with mass spec is perhaps a little simpler because you have a much more limited data set. But I'm curious to sort of larger scale.
Yeah, that's a great question, and we actually were thinking about that. But I think for current stage, since our training data set only contained like 68 glycans, so it might be difficult if we use this model predict the glycans that's out of scope. So maybe not for now to apply to some more complex samples, but this is only the first step. I believe like in the future, we will try to improve our model and apply to more fields.
Oh, great talk. Not sure where I'm facing. So the explainability of your model is very interesting, and you rank your features from important to not so important. Does that mean you can throw away the data from the least important feature and still predict everything else? I mean, have you done that experiment, basically removing data from that seemingly unimportant lectin, and how does it compromise the ability of the model to recreate the problem? And how far can you go? Like, is there basically a stripped down bare bone set of maybe only 3 lectins or 4? Because it's kind of hard to believe, but that would be kind of complementary test of the importance, because I don't understand these sharp values.
Yeah, thank you for the question. That's very interesting. And actually, we have been tested. So I think for the best results, will still remaining some the most accuracy. I tried like 6 lectins, and I think that's the least number of lectins that we could trim down. But for the best results, the 8 lectins were still needed. And yeah, we tried to like delete the least important lectins. Like the prediction will slightly change a bit, but overall is still giving good predictions. But definitely for the choice of lectins, we want to make sure to contain the lectins that could cover most of the glyco features that were in the sample, then the model could have enough information to do the prediction.
Great. Thank you so much, Haining. And that was a really lovely talk.